Thank you for the kind introduction, Dan. And thank you, everyone, for being here, as well as everyone who's calling in or listening in over the line. Today, we're going to be talking about MDS as well as pre-MDS states. And I call it clone wars because really we have been utilizing next-generation sequencing data as well as cytogenetic data to look at how clones battle each other out in these diseases. I have no disclosures that are relevant to this talk. These are some of the other disclosures that I do have. These are a lot of the fun things that we do where we play with in the lab, as well as some of the clinical trials that we run. The overview of today's talk, we're going to start with some challenging classifications of these pre-MDS states. These are the four-letter acronyms that Dan has alluded to. We will go through specifically what I mean by those. Then we'll spend some time on the diagnosis of MDS, specifically how molecular and cytogenetic testing can help us in classification and prognosis of that. And finally, I will cover management of patients with MDS. I know it's a little odd for pathologists to be talking about management of these patients. However, we will be covering transplant outcomes and our roles that we can play in that, as well as new therapies, including immunotherapy, and how pathology plays an integral role in the management of these patients. So advances in MDS since I started here about 7 to 8 years ago is next-generation sequencing started identifying these pre-MDS states, starting with clonal hematopoiesis of indeterminate potential and how that's different from MDS, and then the CUSs, which is idiopathic cytopenia of undetermined significance, how that's different. Moving on to this other term that was thrown around for a while, idiopathic dysplasia of uncertain significance. But really, why do we care? Why should we care about these pre-MDS states? Well, this kind of seminal paper from the New England Journal, which looked at over 17,000 people, actually showed that in the elderly, these clonal mutations are common. Up to 18% in patients over, and these are actually not all patients, they're just people over 90 years old, 18% would have clonal mutations. These are very common genes that we see as well in MDS. These patients ended up having increased risk of hematologic disorders, all-cause mortality, death from anything, coronary heart disease, and ischemic stroke. So it moves outside of the heme realm. Another large study that looked specifically at coronary heart disease and controls showed that in this meta-analysis, where in 2 of these large studies, they looked at whole exome sequencing data. In 3 of these studies, they looked at gene-level analysis. They showed that when you had mutations in DNMT3A, AXSL1, and really, in particular, JAK2, there was a higher hazards ratio associated with coronary heart disease. In the group that had whole exome sequencing, there was a higher odds ratio associated with early onset MI. So this is one of the reasons why we should care. Now, can we do anything about these patients? This is very early preclinical study data where they took some mouse models in this group and they did knockouts with TET2 and produced some knockout models with clonal hematopoiesis here. This is wild type, these are knockouts. And they were able to show that in the wild type mouse versus the knockout mouse, there were increased atherosclerotic plaque growth. This translated to an increased expression of cytokine and inflammation. But furthermore, they also showed that using, a small molecule, MCC950, which is an anti-NLRP3. This is a small molecule inhibitor that reduces IL-1 beta, induced inflammation. They can actually reverse the atherosclerotic plaque growth. But this shouldn't be news to us. Dr. Earl Bendit, back in 1977, had this hypothesis that perhaps atherosclerotic plaques were stemming from one mutated cell. So to summarize this very first section of my talk, the skinny definition of what ICUS, CHIP, and CUS would be. ICUS is idiopathic cytopenia of uncertain significance. These patients would have cytopenias lasting greater than 4 months with no dysplasias, no clonal mutations. Now, in these patients, Their risk of MDS transformation is minimal, but it is not zero, and that is important for us to recognize. CHIP patients have no cytopenias, no dysplasias, but they do have clonal mutations with a low variant allele frequency. And their risk of transformation is low. However, we have just went through some of the other risk factors that these patients may have: MI, coronary artery disease, atherosclerosis. So it may be important to identify these patients. CUSs, these are the clonal cytopenia of uncertain significance. These have been shown in a Mulcavati paper in 2017 to have a hazards ratio for transformation to MDS at about 14. These patients have cytopenias that are lasting greater than 4 months with no dysplasia, but they do have clonal mutations. And this is a little Venn diagram that's a bit confusing, which is why I made this skinny definition table. So mutations that are seen in these different entities are similar, but they are slightly different. So on this side is your CUS patients compared to your MDS. So for a gene like SF3B1, more commonly mutated in your MDS group, however, you can definitely see them in your CUS. In comparison, genes like U2AF1, ETV6, and IDH1, you almost never see them in the CUS group. Now, take this group of patients where they compared CHIP patients against MDS. Genes like BCOR and PEG-A, in comparison to JAK2, JAK2 can be seen in CHIP. Whereas over here, you don't see them in CUSs. So knowing which mutation it is may actually help you. There are some limitations. No gene is mutated in most cases of MDS. No mutation is highly specific for MDS. Clonal hematopoiesis is also common in older individuals, even those without disease. And there's also still limited outcome for these pre-MDS states. It's becoming better. But the conclusion here is that I do think molecular can help diagnose and stratify these patients. ICUS will not have any clonal abnormalities. CUS, mutations of TET2 and DNMT3A are most common, SF3B1 being most frequently mutated in the lower risk MDS. Now, MDS and MPN, JAK2 is almost never seen in CUS, and I'm very careful using the language almost never, because you never say never. In MDS, U2AF1, IDH1, and ETV6 are almost never seen in CUS or CHIP patients. Here's a case vignette. So we now see a 50-year-old woman presents with bleeding, 6-month history of thrombocytopenia, no infection, drug, or history of neoplasia or autoimmune disorder. You have the peripheral blood here. Hemoglobin's okay, white count's okay, platelets is low, and it's been 6 months. Cytogenetics is normal, but molecular does find a clonal abnormality with a fairly low variant allele frequency. You do a marrow, and it's normal. All right, what's the answer? All right, we all learned what these 4-letter acronyms are. So in this second section of my talk, we're going to go through myelodysplastic syndrome proper. This describes a group that is fairly heterogeneous. It's characterized by ineffective hematopoiesis. You have dysplastic features either in the peripheral blood or the marrow. In your myeloid lineage, these include hypolobated or hypogranular neutrophils. You can have abnormal maturation that can be picked up by immunophenotype. In your erythroids, you may have nuclear budding, irregular nuclear contours, or you can have ring sideroblasts. In your megakaryocytes, they can be small, hypolobated, or have separate nuclear lobes. And another very telling characteristic of dysplasia is micromegakaryocytes. These are very small megakaryocytes that are about the size of a myelocyte. You can have abnormal blasts. These could be increased, immunophenotypically abnormal, or have an Auer rod. We prognosticate MDSs based on an international prognostic scoring system, and there's a little R now because this is revised. It is based on the blast count, the cytopenias, and also what karyotype these patients have. In the highest risk group, these patients have a risk to progression of AML within a year. In the very high group, your median survival is less than a year. Lower risk patients can actually undergo monitoring, transfusion, and infection control as support is needed. The minimum diagnostic criteria in 2016 for these patients include findings in the peripheral blood, and the criteria are defined in the WHO as this, but you also have to have at least one of the following. You have to have at least 10% significant dysplasia in your lineage. And you also have to have either or one of these blasts of myeloid phenotype in the marrow, or you have to have a recurrent cytogenetic abnormality such as deletion 5q or monosomy 7. There was renaming of the entities in the 2016 WHO. Instead of previously it was refractory cytopenias of blah, blah, blah. Now we have myelodysplastic syndromes of whatever your morphologic abnormalities is, single lineage dysplasia, multilineage dysplasia, ring sideroblasts, whether they're single lineage versus multilineage. The reason why they separated that category, because these patients commonly have SF3B1 mutations. And the reason why they separated the del5q, these generally do better and have characteristic megakaryocytes. There is also a provisional entity now in the MDS unclassifiables of refractory cytopenia of childhood. And a new category under myeloid neoplasms with germline predispositions. Now RUNX1, ETV6, or GATA2 get their own little category. Our high-risk MDS is challenging. Excess blasts now, if you see any R-rods, can actually be classified here. Cutoffs are similar in the marrow and peripheral blood as previous. Your myelodysplastic syndrome with myeloproliferative neoplasm and ring sideroblasts and thrombocytosis are here now, and your unclassifiables are here. And then you still have your therapy-related myeloid neoplasms, and this could be an MDS or AML. There are important diagnostic overlaps that we must keep in mind as well. We've gone through some of the clonal diseases here, but it is important to keep in mind that there are a host of non-clonal abnormalities that is also identified that can overlap with MDS. Medications, infections, autoimmune disorders, systemic diseases, alcohol abuse, and deficiencies in vitamin or some of these essential metals. And so when you're making a diagnosis of these pre-MDS states as well as MDS, it is important to keep these in mind and to make sure you rule these out as well. There are molecular overlaps as you make your diagnosis of MDS as well. There's overlap between your MDS propers, your MDS with MPN overlap, your MPN diseases over here. And as you can see, none of these rings are separate. They all overlap with each other because the mutations in the genes are not specific. So our conclusion for this section is that proven clonality in MDS can help define and prognosticate. The cons of relying on molecular biomarkers to define your MDS is that somatic mutations lack both sensitivity and specificity for any one entity. TET2 and SF3B1 are seen in about 25 to 33% of MDS only. ASXL1, SRSF2, DNMT3A are in about 10 to 20%. But there are also pros here. We are beginning to understand that mutations are specific for certain entities. So mutations in U2AF1, RUNX1, TP53 can help. And with the germline predisposition syndromes, if we see germline mutations in RUNX1, ETV6, or GATA2, those can also help us with our management of our patients. So in this last section of today's talk, I want to talk about how do you treat these diseases? And it's kind of funny having a pathologist talk about management, right? But there really wasn't much in terms of management and therapy for MDS. In pre-MDS states, really it's a watch and wait. What do we do? There are some kind of acknowledgments now that perhaps we may need to do something, but still currently there is no established treatment for pre-MDS patients. For MDS patients, for patients with MDS del5q, you can treat with EPO and possibly with lenalidomide after that. DNMT3 inhibitors, azacitidine and decitabine. These were FDA approved in 2004 and 2006. These were based on 2 trials, the CALGB phase 3 trial for azacitidine, where they showed 60% response. And subsequent to that, the decitabine was approved based on a 17% response rate in very high-risk MDS patients. However, despite all of these advances, transplant is truly the only curative option for MDS still. But transplant is not the best answer still. Relapse is still the major cause of post-transplant death. This is CIBMTR data for autologous, HLA matched SIBs, and unrelated donors. As you can see, that big blue pie on each of these is primary disease. These patients are still dying from relapse of their initial disease. So why are these patients relapsing? And what can we do to fix it? A study by Jang Hee, this is Dr. Wu over at the Hutch, he tried to use azacitidine as a single agent after transplant relapse to see if he can actually treat these patients and get remission. He was actually able to get a 30% response rate, and so thought it's possible. But this is still pretty crappy, I would say. Can we do better? Can we? We have all of these tools in pathology. Can we use it to help us understand this problem? We did a number of studies. We looked at the relapse after all of our allogeneic transplants for MDS. And when we reviewed, we had 973 MDS patients that had more than 18 months of follow-up. And of these patients, 408 patients were alive without relapse at 18 months. So we took those patients, and of those, 254 patients relapsed. So it's more than half. From there, what we did was we plotted them on this cumulative incidence of relapse, and really the 18-month cutoff was just where the inflection happened, and we arbitrarily said, okay, let's compare the people who relapsed before 18 months to the people who relapsed after, and see if there's any difference. Why are they doing this? And 213 patients relapsed before 18 months and 41 relapsed after, and we looked at what is the difference between these two groups. One of the things that we did have to do was go back and perform CGAT testing on all of these patients. That's chromosome genomic array testing. We already had karyotype data, morphology data, flow data on these patients. And so with this testing, we were able to look at the whole genome. The pros is that we can now detect small deletions and duplications that we could not see by karyotyping. We were able to detect loss of heterozygosity. The cons were it was relatively an expensive technique, and we could not detect balanced translocations, but we had that data from the karyotype. So when we did chromosomal microarray at SCCA, this was done with an Affymetric cytoscan chip with 2.5 million probes. The cutoffs that we used in our assay was 100 kb for copy number alterations and 100 megabases for copy neutral loss of heterozygosity. Typically, our data does not look like this. I just used the pretty colors for this talk. Typically, you would have a monocolor, so it's easier to see the aberrations. So on this top line, as you're reading this, the copy number probes in 1Q, you can see, for example, here, is up, meaning there is a gain in 1Q. For your SNP probe, you should have 3 tracks in a normal genome. In this middle line here, you can see in 4Q, you have loss of your heterozygous track, so therefore loss of heterozygosity. What we saw was that survival was poor in the relapsed patients, regardless of early or late, not surprising. We further looked at if there were any clonal evolution, any changes in karyotype, based on whether they were early or late. And really, we didn't really notice a huge difference. A third of these, we really couldn't go back and do everything. On about a third of these really had no change, which was a surprise. We would expect that patients who had late relapse would have some sort of clonal evolution, but really we didn't even see that. But a third of these patients did have clonal evolution. So the transplant outcome in MDS is that clonal evolution is seen in a portion, about 32 to 41% of these patients. The risk of relapse was highest in MDS that has transformed to AML, and lowest in patients that had chronic graft versus host disease. Very poor risk cytogenetics were more likely to have early relapse, and that we really needed to use both cytogenetic and CGAD data to help track the clones at relapse for the study. But there were still unanswered questions. Were these late relapses donor-derived? Were they therapy-related? We had more work to do at this point. And what's really interesting, those folks over at WashU, the other UW, they did this really interesting study. This was published on Halloween, just about, 14 days ago? They did a retrospective analysis. They paired relapse patients, 15 relapse from transplant, 20 relapse from chemo. They looked at the diagnostic sample and the relapse sample. They did exome sequencing as well as single-cell RNA sequencing of immune-related genes. And what they found here is, this is one of the figures. And so these are the HLA genes. What they found as they compared the diagnostic sample to the relapse sample was that there was a decrease in the expression of these HLA markers in the relapse samples for the transplant cases, whereas in the chemotherapy cases, that loss of expression on these HLA markers wasn't there. Could there be an immune escape mechanism that is in these transplant relapses that perhaps isn't seen in the chemotherapy cases? So this actually was very interesting, and they thought that perhaps it's a downregulation of these MHC class II genes in the transplant patients. And so we have work to do. Perhaps we need to go back to our MDS cases and do this study. So following up on our case, we actually noticed from our CGAT data that a high number of our patients had copy-neutral loss of heterozygosity. And we wanted to see what that impact was on survival in our MDS patients. We went back, we looked at sequential cases over 5 years and found 69 patients that had CGAT data in SCCA. We reviewed cytogenetic, morphology, immunophenotype, as well as compared that to their clinical outcomes. Our hypothesis here was that the MDS cases with CNLOH region-specific abnormalities may have specific morphologic and immunophenotypic aberrancies that can impact survival. So we've already gone through how to do a CGAT. Now, I'm going to go through briefly how to calculate total genomic aberration scores. Here, we take that same CGAT data. We take the number of megabases that are abnormal in the genome, including both copy number alterations as well as copy neutral loss of heterozygosity. We give it a score. We add it up. The summary of our findings in our 69 patients is that, luckily, this actually represents MDS patients very well. A lot of our patients had that 5q deletion, monosomy 7, trisomy 8. Okay, we're good. We're doing good with our diagnoses. However, interestingly, the CNLOH findings in these patients was that very commonly we had CNLOH in 4q, in 7, 9, 11, 17. And these corresponded to the genes such as TET2, EZH2, JAK2, CBL, and TP53. Our median TGA in this study was 70. When we compared the CGAT data versus metaphase cytogenetics, we were able to detect additional abnormalities on normal metaphase cytogenetics. These include both patients with just CNLOH with nothing else, as well as small indels. We were also able to further clarify a couple of cases that had failed culture. But most significant from this study was that we were able to show survival differences based on the total genomic aberration score. When we took all patients and separated them according to the median TGA value, there was a significance in survival differences. However, most useful perhaps is that when we separated this based only on the IPSSR high-risk patients, so these are patients that are already deemed to be doing very poorly, we were able to show that patients in the high-risk category, if we had a lower TGA, tended to do better. So the other thing we noticed in this study was that the most common CNLOH regions were 9p and 17p. And in the 9p CNLOH regions, we had 8 patients. 7 of these actually had oncoplex data. And of those 7, 5 of them actually had the V617F mutation. 2 were negative. 1 actually was untested, and that's because we couldn't get any more DNA from any source for that patient. In our 17p CNLOH, these were more commonly associated with multilineage dysplasia. So our conclusion from this study was that CGAT for MDS is now a part of our pathways at SCCA and has become very integral in the diagnosis of MDS. TGA values show differences in survival. The presence of CNLOH and copy number alterations are important and should be used in the calculation of risk scores for MDS. NGS data is synergistic with CGAT, but the number of cases in this study was still comparatively low. The potential use for targeted therapy when we do discover CNLOH in specific regions, such as JAK2 and EZH2, may be important. So now I want to spend a couple of minutes here reviewing immune therapy for MDS and why it might work. So leukemias were one of the first cancers to be cured by transplant, which is somewhat of an immune-related mechanism, graft versus leukemia effect. Leukemic cells express checkpoint inhibitors. Key ligands include PD-L1, CD80, CD86. Immune evasion occurs secondary to the depletion of leukemic antigen-specific T cells. Currently available for AML and MDS include checkpoint inhibitors, multiple trials, in PD-1, PD-L1, CTLA-4, in combination or not with hypomethylation agents. There are CD123 CAR T cells that are in development, along with others. WT1 TCR CTLs, which I will be going over in a little bit. This is a trial that's ongoing at the Hutch. So this is a review paper that describes the emerging role of checkpoint inhibitors for use in AML and MDS. In here, it describes 6 phase 1/2 clinical trials, and this is as of April this year. I would imagine that a couple more trials has probably opened, and these specifically target PD-1, PD-L1, and CTLA-4. When we look at the engineered T cells, we have chimeric antigen receptor versus T cell receptor cytotoxic T lymphocytes. As I will be showing examples of both, I want to describe briefly what the differences are. In a chimeric antigen T cell, you have your CAR expressed on the surface of your T cell that recognizes a specific surface protein on your cancer cell. In comparison, on a TCR T cell, you have your gene-modified T cell receptor that recognizes an HLA peptide complex, which then can actually get internalized into your cancer cell and therefore be processed and recognize a target protein that is inside your cancer cell. This is a key point as to which tool you will use when you are fighting your cancer. For CAR T cell therapy, you start by removing blood from a patient to get to your T cells. You make your CARs in a lab by inserting a gene for your chimeric antigen receptor. And as that takes, your T cells will express the CARs on the surface. You expand those, and then you have your living drug, which then gets infused into your patient. And why we think CD123 may be a good target for CAR T cells is that it is overexpressed in the stem cells of high-grade MDS and AML. This shows low-grade MDS, high-grade MDS, and AML. Of course, it's most highly expressed in AML, but as you can see, there's a graduated expression level here. In a preclinical model of CD123 CAR Ts for MDS, these are cultured patient cells where they took a mock CAR versus a CAR 123 specific T cell and did culturing with two patient samples. One, number 388, they measured this after 48 hours of exposure. Number 833, they measured it 72 hours after exposure. And as you can see, the number of CD123 MDS cells is almost zero. It's not exactly zero, but it's almost zero compared to the mock. Interestingly, the cytokine protection, very similar to the CD19 CARs, was also high. So a potential for cytokine release syndrome there as well. When you compare this in a PDX mouse model, the same thing happens, where in comparison to the mock, you have a dramatic drop in your CD123 MDS tumor cells. And when you take that same PDX mouse and look at the marrow, you have emptying of the marrow MDS cells. But this is preclinical study. These are cultured cells and a mouse PDX model. So next, I want to talk about when you look at T-cell receptor generated gene-modified T cells. So here you have a matched HLA donor. You really want to select for virus-specific T cells because we've identified that these typically have increased frequency of central memory T cells, as well as improved persistence and reduced alloimmunity, which are alloreactivity, which is good for your product. You would then transduce a TCR into these patients that is specific for the WT1. And what you want here, so as you select for your virus-specific T cells, then you want to transduce this little green thing over here, which recognizes your WT1 epitope. You further use going through a GMP flow sorting, you select for these T cells that are modified and selected to target what you want. In our case, it's WT1. And then you expand it similarly. And before you infuse it into the patient, you have a live drug. The first protocol that we got involved in pathology was Fred Hutch protocol 2498. And this was initially developed by Phil Greenberg. Subsequently, he recruited Aude Chapuis to take this on. So Will's tumor was overexpressed in poor prognosis AML, CML, and MDS, and is a nuclear marker. Thus, this is the reason why CARs don't work, and we needed to go after them with these immune gene-modified T cells. So an overview of the initial study was that they had two arms, prophylactic arm and treatment arm, high-risk patients after HCT, and I will tell you that to date, the prophylactic arm, nobody's relapsed, so it's great. The treatment arm is patients who relapsed after transplant, then gets put on this protocol. However, by the time pathology got involved, it was kind of late and was retrospective. They didn't select for WT1 expression before this study started. And what we did was retrospectively, we went back and looked at WT1 expression on all of these cases, did RT-PCR as well as IHC. We tried to do ISH, but that's another story. There was 25 patients at that time. We have actually since then reopened 2498 and are enrolling more patients. We performed quantitative RT-PCR as well as immunohistochemistry. And then subsequent to that, based on this data, we actually figured out that WT1 isn't highly expressed on every single AML. And we figured out also that you need the WT1 expression for efficacy. And so 9296 was subsequently written with a prospective evaluation for WT1. And now WT1, either immunohistochemistry or RT-PCR overexpression, is required for eligibility into the second trial. So the conclusion here is that immunotherapy for MDS is promising. Like I said, for 2498, in the first arm, nobody's relapsed so far. In the second arm, some people have, and that is why we've based the second trial, 9296, on a pre-enrollment WT1 expression. And then, so the future therapies can be improved by actually looking at multiple targets and perhaps a combination of therapy as well as immune therapy and specific targeted agents. But MDS AML is a heterogeneous disease, and possibly debulking is important. Some of the future directions that we would like to do in our lab would be single-cell sequencing, specifically with some of these immune therapy treatments, and as well as the MDS study now that we have that new study coming out. We really want to go back and look at that relapse study. The tumor microenvironment in MDS is also extremely fascinating. Now with multiplex immunohistochemistry that is available, we want to take a look at that. And also a couple of translational medicine and biomarkers work that we're doing. This is a lot of the clinical trials that we're doing with SWOG, as well as immunotherapy studies. And some HDAC inhibitor trials are also coming down the pipe. But the acknowledgements are most important. Our patients and their clinical teams, none of this would be possible without our patients and the donation of their samples. The Radich lab and the molecular oncology lab has done most of this work, as well as cytogenetics in the FANG lab. Biostats, Greenberg, Chapuis with all the immune therapy work. The MDS research group that I'm a part of, and Fred Hutch Pathology, where a lot of these transplant on AML samples are from. Genomic score and HemePath, of course. Questions?
So that was very interesting. I work on microbes, so there's a lot of parallels to making vaccines against other foreign things. My question was, you said there's class 2 downregulation in that Washington University study. Class 1 downregulation seems more relevant to what we're talking about. So in the Wilms tumor, for instance, is it sufficient to just measure the expression of the antigen, or do you really need to measure the presentation of the antigen, which is a combination of how much antigen and how much, class 1? Is that something you consider, or does the antigen expression tell you everything you need to know?
So I took one picture from the whole paper. So there's actually functional studies in that paper as well. I actually have the paper if you want to see the full thing. So that is just one tiny little snippet so I can fit it into one slide. But yes, absolutely is it important to do functional studies in addition to looking at MHC class 2. And for that, they actually looked at an entire immune repertoire. So they didn't just look at MHC class 2. Although in their conclusion, they identified that as one of the most significant factors.
Again, interesting work. The recognition that there's a lot of immune response in MDS is intriguing and raises the question of why there are cytopenias in patients with MDS. It's always a kind of a mystery. It's not a packed marrow. There seems like a lot of the precursor cells are there. Is there thought to be a role of autoimmunity, perhaps due to epitope spreading, that leads to the cytopenias of the normal cells?
That is a very difficult question. And I think that is also the crux of all of the investigation that's going on with the pre-MDS states and actually looking at the autoimmune diseases. And there is actually a number of literature and papers that specifically addresses autoimmune diseases, ITP, et cetera, and Sjogren's, and looking at their connection with pre-MDS states as well as development of MDS.
So I have a question for you about the clonal cytopenias. I mean, you made it pretty clear in your talk that these pre-MDS syndromes, there are lots of different mutations and there's lots of overlap between different pathological categorizations, I'll call it, to simplify. So my question is, are there particular mutations in these clonal cytopenias that end up providing a specific cytopenia?
So that is a good question. We have identified that perhaps JAK2 may be associated with, say, myelofibrosis and maybe megakaryocytic abnormalities, but no, there isn't anything that is absolutely specific. Perhaps SF3B1, maybe with erythroids, but I say that with this major grain of salt because really we're just beginning to understand some of these lineage-specific mutations. And yes, but no. So this is part of the I cannot be specific about these, but there is some inferences.
So just sort of expanding on that idea, based on what people have studied over the years in terms of hematopoiesis in the elderly, how does the finding now of these mutations in blood cells in elderly people, sort of relate to the original findings of hematopoiesis in aging people? And also, do you want to speculate on how these mutations might in some way relate to the development of cardiovascular disease?
Yes, actually. So there's a hidden slide that I have in here that I didn't show. No, so TET2 is one thing, for instance. There's actually a number of papers that I didn't end up adding in here. So people are now beginning to look at these inflammatory states that these pre-clonal hematopoiesis mutations are perhaps giving you. And the cytokine and immune repertoire that are putting you in this pro-inflammatory state that really Dr. Bendit had noted in the very beginning that puts you in this kind of cytokine rich environment, and perhaps that is what's causing the MI, atherosclerosis, arterial diseases, cardiovascular disease, kind of pro-inflammatory state. So it really is kind of all beginning to tie together. And so this is an exciting area right now that we should look into, and it relates all to the vasculature and the inflammation, cytokine storm, immune regulation. So.
All right, well, thank you, Dr.
Thank
Yeung.
you.